All cells they produce from bone marrow, even the B cells still produce from bone marrow. The important thing about the B cell maturation also inside the bone marrow. So B cell mature where B cell not going to thymus. This is the difference from the T cell. Okay. So acquired immunity having the two parts, one T cell and B cell. The T cell they produce from bone marrow, they enter to thymus, okay, for further processing. Yeah. So how about the B cell? The B cell they stay inside the bone marrow and their further processes, further all processes going inside the bone marrow. Okay. The T cell they directly go to thymus, but the B cell stay inside the bone marrow. Clear? So B cell, when they leave the bone marrow, they're going to lymph nodes. How about the T cell? T cell first enter to thymus, then their thymic education there. After passing through the thymus, then going to lymph nodes. Okay. B cell after the bone marrow, they're directly going to lymph node. Clear? This is the main difference from the T cell maturation and B cell maturation. So every aspect of B cell maturation we already done. Okay. Negative selection, positive selection. So, but the B cell maturation inside the bone marrow, not inside the thymus. How the B cell recognize the antigen? How the B cell recognize the antigen? They receive antigen B cell. Do you remember about the T cell receptor? T cell they having how they recognize the receptor. They recognize the antigen with the help of T cell receptor. T cell receptor. This T cell receptor interact with MHC protein. MHC protein, clear? On the B cell, the B cell, they not have receptor like that, similar receptor. Here we having a receptor IgM and receptor IgD. So these two antibody, they act like B cell receptor. Another difference here, okay? What are the B cell receptors? B cell receptor IgM, B cell receptor IgD. So these two antibody they act like B cell receptor. Is totally different from T cell receptor. T cell receptor is a specialized separate receptor. Okay. So B cell receptor they recognize the antigen. So antigen coming from outside, antigen coming from outside. You see the difference here. So these are not proteins, okay? The antigen coming from outside. If they polysaccharide in nature, the DNA particles, some small molecules. So antigen, if antigen, in one class of antigen, we already discussed about what is the property of antigen. The antigen, much bigger antigen, more immunogenic, yeah. The mostly antigen, they are protein in nature, okay? They are protein in nature. So they have dual property of immunogenicity and also immunoreactivity. So if the antigen nature polysaccharide, antigen nature nucleic acid, or antigen are very small in size. So all the small size antigen are directly by B cell. So now they attach with the receptor of the B cell, IgM and IgD. Clear? IgM and IgD. So now antigen already received by B cell. Now what the B cell is doing? There are two pathway here. The B cell take this antigen into inside. Okay. Now this antigen inside the B cell. This B cell give antigen to T cell. This B cell give antigen to T cell. How B cell give antigen to T cell? What is that protein? Do you remember MSC2? B cell using MSC2 protein interact with T cell receptor. Okay. Now the antigen presented to T cell. B cell give message to T cell. Uh, which T cell? CD4. Which T cell? CD4. Okay. So this CD4 T cell, the further process done by T cell. Clear? The same like macrophage, same like dendritic cell. Can you tell me what is the secondary signal here? If B cell present antigen to T cell, what is the secondary signal? It's a primary signal which we know. What is the secondary signal here? What must be secondary signal?
B7 protein.
No. B7 to CD28, that's for macrophage dendritic cell.
Oh.
Secondary signal here I already explained in that lecture antigen, okay? That's a T cell activation there. CD40L and CD40. So this must be the secondary signal here. Here I can't use B7, okay? No B7 here, CD28. So this just for macrophage. If macrophage present antigen to T cell, then secondary signal B20 is B7, okay, and CD28. And if the dendritic cell present antigen to T cell, the same secondary signal. But in case of B cell, now you can see the B cell receive antigen, okay, directly receive antigen. What are the B cell receptor? They receive antigen with the help of IgM and IgD. Clear? So these two antibody, they act like B cell receptor. Understand? They act like B cell receptor. Now antigen presented to T cell. Antigen presented to T cell. How they present? Same mechanism like macrophage. Okay. So now secondary signal. So what happened now? Activation of T cell. Activation of T cell. This you know already. Okay. Another way, when directly antigen come to the B cell, what's another way? Another way here, the T cell, this B cell, this B cell, if not present this antigen, what happen? This B cell self-activate. This B cell just take antigen inside and self-activation and self-activation. If self-activation there, if self-activation there, so B cell activate by itself, independent, not take help from T cell. Okay. If taking help from T cell, I must present antigen to T cell, and then T cell give more power to B cell for fighting. Okay. If the B cell don't want to present this antigen, if the B cell don't want to present this antigen to T cell, now this antigen already inside the B cell become active. So B cell start producing antibody. Which antibody you just can produce? IgM. Because B cell not taking any help from T cell. Clear? B cell, if not take any help from T cell, if just B cell want to handle by its own way, so B cell activation without T cell have minimum power, have minimum ability to produce antibody, they're just able to produce IgM, just able to produce IgM. Clear? Okay. If the B cell proper way, B cell give message to T cell, then T cell give power to B cell to produce all five types of antibody can be produced: IgM, IgD, IgA, IgE. It depends upon what type of antigen. Clear? The difference clear or not? So if the antigen directly taken up by B cell, then the more power the T cell independent antigen, T cell dependent antigen. Sorry for some interruptions with throat problem. T cell independent antigen. So now you understand about what are the T cell independent antigen. When antigen directly going to B cell and the B cell activation start. So the signal is not given to T cell. B cell start working by its own. Okay. B cell start working by its own. Then the B cell only able to produce one antibody, only able to produce only one antibody, that's IgM. Okay. So the class switching power, let we see in antibody section, the class switching power is not much. The B cell not much having the class switching power, not able to produce other category of antibodies. Clear? So this is a difference between if the B cell activate by itself, okay, by itself, not take help from T cell. But the normal way of immune system activation, all the antigen presenting cell, they must take help from CD4 cell. Clear? They must take help from CD4, then they can be active properly. Clear? Because CD4 cell is your key cell in your immunity. So if the antigen is presented to T cell, then the proper activation of B cell, the B cell full activation, the B cell full activation. Okay. Okay, let's see what's the maturation here. It's a lymphoid mother cell here. It changed to pro cell. It's a pro B cell. It's a progenitor here. Okay, they're having Ig heavy chain. Antibody, antibody having two types of chain. One heavy chain that we see in antibody section. Okay, heavy chain and light chain. The two chains here. Two heavy chain, two light chain. Okay, so the heavy chain rearrangement inside the pro B cell. Next, this B cell change to pre-B cell. Pre-B cell, now you can see light chain gene rearrangement there. So in pre-B cell, we're having light chain. All these are cells, they are still immature. Pro-B cell, pre-B cell, then change to, or is now immature B cell, okay? So at the surface, what we have at the surface? B cell receptor IgM. When it changed to, when it changed to mature B cell, this mature B cell now having both IgM and IgD on the surface. I already explained these are the receptors, okay? These are the B cell receptor, IgM and IgD. What are the B cell receptor? B cell receptor. So we having IgM and IgG, IgD. So these two antibodies, they act like B cell receptor, okay? Now you can see the what are the steps of maturation of B cell. So every step inside the bone marrow, okay, you can see here. Immature till here, till immature cell. This immature cell now enter to blood, now going to lymph node, they change to mature B cell, and the mature B cell become active. When they become active, they change to plasma cell. They change to plasma cell. So active plasma cell, they producing what? They producing antibody. And one cell after activation change to memory cell, just to memorize the infective agent. Okay, just to memorize the infective agent. So these all are the steps for activation of B cell. So you can see the mostly maturation inside the bone marrow, inside the bone marrow. Clear? The immature pro B cell, pre B cell, immature B cell, when they exit the bone marrow, they become mature. Now going to lymph node, they become their active. When they become active, they're producing, you can see mature B cell, they have B cell receptor. So B cell, this B cell able to recognize antigen, able to recognize antigen. Clear? So this antigen recognized by mature B cell can given to T cell, also can self activation there. If this antigen presented to in proper way, presented to T cell, then B cell have more power to produce all types of antibody. If this antigen is self-handled by the B cell, is self-controlled by the B cell, then the B cell not able to produce all classes. Clear? Only able to produce one antibody, IgM. Just can fight against acute infection, not ability to fight against chronic infection. Understand? So it's that we already described about the types of antigen. Some antigen just handled by B cell, they're not presented to T cells. Okay, so these are thymus independent antigens. Thymus independent mean T cell. T cell belongs to thymus. Okay, and if the antigen in proper way presented to T cell with a like from the epithelial cell here, this is the third number epithelial cell, B cell, okay, macrophage, dendritic, and B cell. So this antigen presented to T cell, then we have more chance for stronger immunity. Understand? This is a difference. Clear? Okay, let's see the another cell here. B cell, T cell done. Was a natural killer cell. Natural killer cell. So natural killer cell, these are also lymphocytes. These are also lymphocytes. Okay, they're not passing through thymus. Of course, they're not passing through thymus. Okay, they don't interact with the antigen. They just have, you can see, they not have CD4 protein, they not have CD8 protein. They are direct killer, okay? They just recognize the cell, recognize and kill the target cell, especially virus infected cell, especially tumor cells. Clear? So they don't need any presentation of antigen. It's mean. So that's why they don't need activation. They already active. So natural killer cell is also part of innate immunity. Okay, they just know the fighting. They don't know how to fight. They just know the fight. Find and recognize and kill. Clear? Find and recognize and kill. Oh, it's a foreigner, just kill them. So these are the nature killer cell. So nature killer cell, they don't want any antigen presentation. They understand? They not present to others. They not receive from others. They just directly go and recognize the antigen and just start fighting. And just start fighting. Okay? Clear? Selection done. This is selection already done. Okay? Positive selection, negative selection. Positive selection, negative selection. Bad receptor, negative selection. So I'll explain, okay? What's a positive selection? Now I describe what's mean positive selection. You know, every T cell have receptor. Yes, every T cell have receptor. Of binding, if the receptor bind with antigen affinity, little bit low affinity, we receive positive antigen. Receive positive selection. Okay? And also able to give message to inside. For example, this antigen bind with T cell receptor. Antigen bind with T cell receptor, and that this receptor give message inside the DNA of T cell. Okay, so if the cell have low affinity for antigen, one thing, low affinity. Low affinity here means the binding not must be much stronger. Okay, if the so these cells are positively selected. What's mean negative selection? Negative selection. If they not bind, if they fail to recognize, if they not they don't want to bind with MSC protein, they don't want to detect the antigen. Okay, so if they not bind, they're negatively selected. If they bind, much strong binding is also not needed. Okay, this is the antigen presenting cell. Okay, antigen presenting cell having what protein? MHC protein. MSc protein. Okay, this is a T cell. Are getting education inside the thymic school. Okay, the CD4 or CD8. CD4 or CD8. Okay, clear. So they detect. They having what? They have T cell receptor. Which cell must be selected for immunity? Which cell not be selected for immunity? What is the education here? This T cell we teach this receptor first. This is a receptor, okay? Receptor present on T cell. This receptor must bind with MSC protein. This is one condition for poster selection. This is, it's according to merit. What is the merit for your selection? If you want to be part of the fence, first thing, you must bind with MSC protein. Clear? How I bind? A must be weak binding. Understand? Just receiving, not hold MSC protein for a long time. Just receive antigen and left that MSC protein. Understand? First thing, the first condition for poster selection to be part of the fence, T cell receptor must bind with first condition, must bind with MSC protein. The cell T cell receptor must bind with MSC protein. Okay. How to bind? Weak, strong, very strong, weakly bind with MSC protein. If the T cell receptor have this property, that cell must be our immunity part. That's the poster selection. Understand? That's the poster selection. Okay. What's mean negative selection? If the T cell receptor not bind with MSC protein, can you select that cell for immunity? Can you select that cell if that cell don't want to recognize antigen? don't want to attach with MSC protein. So if the cell T cell receptor not able to attach with the MSC protein, how can detect antigen? So that T cell is useless. That's why it's a negative selection. Understand? So that cell is negatively selected cell. That cell is negatively selected cell. Clear? So fail to bind with another thing, like if the too strong binding, if the too strong binding, if they are too strong binding, like they attach with MSC protein and induce apoptosis of this. Non-A too strong binding is also very important. Okay, so these two property, if they not shake hand with T cell receptor, not bind with MSC protein, that cell must be negatively selected. If the very strong bond with MSC protein is also not good thing. Okay, it's also not good thing. I'm teaching. I'm a thymus. I am teaching to my T cell. Okay, T cell. You have receptor on your surface. Yes. Okay. You how to use this receptor? You must bind with MSC protein. I am teaching them. Okay. The thymus teaching to T cell. How they teaching? Thymus teach them. Thymus now taught them what education here. T cell receptor. Oh, the teaching T cell receptor. You use your T cell receptor to detect antigen. How they detect antigen? You must bind with MSC protein. This is a first condition. This is a merit. Okay, the first merit. If you want to be a part of body defense system, body military system, you must bind with MSC protein. Clear? This is a first requirement for post selection. If you not fulfill this requirement, you must be degraded. Okay, you must be degraded. You will not selected for the immunity defense system. You're not supposed to for immunity defense system. You negatively selected and you be killed by other immune cells. Understand? Clear or not? Okay, supposed to selection way. And the second property, how to bind. First thing, must bind. Second property, you must shake hand with MSC protein. I am teaching to T cell. Okay, I am teaching to T cell now. That you must shake hand with MSC protein softly. Must shake hand with MSC protein, receive antigen, do this action very softly, very gently, okay? Not bind MSC protein very tight bond there. If the very tight bond, there must be, oh, they start a process there. It's not a good thing. So, too strong binding is also not allowed for T cell. If the T cell not bind with the like MSC protein, the receptor not bind with MSC protein. So these two negative criteria here. Okay, if no binding, that cell must be negatively selected. If they don't want bind, I'm teaching that you must bind. Or they're saying T cell saying I don't want to bind with MSC protein. If they're not attached with MSC protein, they're negatively selected. If they're negatively selected, they must be killed by other immune system cell because they can start autoimmune disease. Clear? The criteria understand or not? What's the criteria for negative and positive selection? What's the criteria for negative and positive selection? The negative positive selection must bind. Okay. Positive selection must bind. I just make it easy. Must bind. How to bind? Bind weekly. Bind weekly. Must bind MSC protein. Bind weekly with MSC protein. If you fulfill this criteria, it's a criteria for postal selection. Okay. If you complete this criteria, you will be selected for immunity. Understand? You will be selected for immunity. You will be selected for immunity. Must bind and bind weekly with low affinity. Okay. What is the criteria for negative selection? The cells not selected for defense. They don't bind. Their T cells have to don't bind with MS protein. That cell must be negatively selected. Okay. If they bind, first they say I don't want bind. If they bind too strong, too strong bonding with MSC, too strong MSC binding. This is also not good. So if the cell having this property don't want to bind with MSC protein, if bind and bind too strong, so that cell must be negatively selected. Understand? So negatively selected cell must be killed by positively selected cell. Otherwise, what happens? Otherwise, autoimmune disease will start. Okay, this about negative positive. What's mean homing? After the maturation, B cell and T cell. B cell mature in bone marrow. T cell mature in thymus. After maturation, after their complete selection done, okay, selection done. So from the bone marrow and the thymus, now the cell are mature. These are mature. What is the meaning of nave cell? What is nave cell? Nave mean they are mature, but they are inactive. Okay, nave word is opposite to active. Nave mean inactive. Nave mean inactive. Active is active. Okay, so the nave lymphocytes, they're ready to begin their role. They're ready to begin for defense. Okay, understand? Now these mature, these are the mature cells. They are educated cells. Okay, mature B cell, T cell, but these are naive cell. Understand? Mature inactive cell now circulate in the body. Clear? They're moving throughout the body, and they're living in. They're moving where? They're going to lymph node. They're going to lymph node. This is the homing process. Understand? After maturation, they circulate throughout the body, and they what's the residency place? Lymph nodes. They're going to lymphoid tissue. Okay. This is the lymph node structure here. Let's see what's the lymph node structure. This the outer part is cortex. Okay. This the inner part is the paracortical area. This one is paracortical area. This deep part is medulla. This deep part is medulla. So what cell present in cortex, what in paracortex, what in medulla? So are different. You see, this is a full, for example, this is the headquarter, yeah? Headquarter of T cells. Headquarter of defense cells. Which cell living where? In the cortex. In the cortex, we're having follicles. You can see the yellow color structure here. So these are the follicles. So inside the follicle, cortical area, all B cell living here. All the B cell, they're living here. Clear? All the B cell. So these are the follicles. So in the area, this area, I shared with the black color. This is the paracortical area. This is the paracortical area. So T cell. So this area full of T cell. This area full of T cell. So T cell living here. What is the medulla? In the medulla, we having macrophage. In the medulla, we having mast cell. In the medulla, we having neutrophil. In the medulla, we having basophil. So all other body cells, all other defensive cells, they live here. Okay, macrophage, basophil, xenophil. So like this. Okay, all other cells here. This is the distribution here. So in how they're living inside the lymph node. The B cell living in follicle. Okay, so which area? Cortex. So we can set cortex for B cell. The area close to cortex, T cells. An area deep in lymph node like medulla for macrophage and for neutrophils. Clear? Understand or not? Okay. It's this their home, how they're living inside the lymph node. What's the spleen? So inside the spleen, inside spleen we having white pulp and red pulp. White pulp for WBC, okay. Red pulp we having red blood cells there. So in the white pulp, what we see in the white pulp? This is the vessel here, okay. This is the artery. This is full is a white pulp, okay. This is full is a white pulp area. So in this is the artery here. It's one arteriole. Around the arteriole, can you see this purple color here? This is a sheath, purple color. You can see this purple color, yeah. So this purple color is a periarteriolar, periarteriolar lymphoid sheath. Periarteriolar lymphoid sheath. Okay. So in this periarterial lymphoid sheath, we're having lot of lymphocytes. Understand? Follicles for lymphocytes. Macrophage is also close to it. Clear? Macrophage is also close to it. So in this sheet we having lymphocytes here. So what is the area for lymphocyte in spleen? Where is inside the spleen? Inside the spleen White blood cell present in white pulp. Which area of the white pulp? So white pulp you can see having very big area white pulp here. So we also have macrophage there. We also have neutrophils there. Clear? So exact area of lymphocyte around the arteriole. Around the arteriole. Around this arteriole we having lymphoid sheath. Which sheath? Lymphoid sheath. Peri-arteriolar lymphoid sheet. Okay, in this sheet we having T cells there. Clear? How they working inside the spleen here? Naive T cell migrate to cortical area of spleen. Naive B cell also responds to some chemicals there. Okay, so all these lymphocytes inside the spleen they present where? Naive T cell, what's the home? Peri-arteriolar lymphoid sheath. Peri-arteriolar lymphoid sheath. Okay, clear or not? Peri-arteriolar lymphoid sheath. So this is a location of T cells and B cell inside the spleen. Okay. So homing done, selection done, B cell intro done, T cell activation done. This is everything about B cell and T cell. Okay. So their maturation, their activation, how they working. So let's see what are the other body cells. So what are the other body cells? The cells of immune system. We having like lymphocytes there. This we done. Very important cell for acquired immunity. We having natural killer cell also there. Okay. So myeloid progenitor cell. Myeloid. There are two main lines there. So myeloid cells, for example, this is a mother cell. Okay. Pluripotential hematopoietic stem cells. Pluripotential hematopoietic PHSC. This cell changed to two lines. Okay. One lymphoid, here I can write here, one lymphoid line and other myeloid line, lymphoid and myeloid line. So in lymphoid line, it's also very helpful for you to understand the blood cancer, okay, the different type of blood tumor. Lymphoid line change to T cell, lymphoid line change to B cell, lymphoid line form natural killer cell, okay. So these are all lymphocytes. And myeloid line, from myeloid monocytes, from myeloid line basophil, myeloid line neutrophil, myeloid line neutrophil, basophil, eosinophil, red blood cells, platelets. Okay, so all these cells formed by myeloid line. Understand? This is about their origin. Okay, so myeloid progenitor cells here. Let's say myeloid progenitor cells. Let's look. Lymphoid progenitor cell we already done. Okay, everything done about T cell and B cell. Let's see some important properties of other cells. Granulocytes and agranulocytes. We divide into two groups. Okay, some cells they having granules inside. Some cells they having granules inside. Some cells they not having granule inside. If they are granulocytes, they having granules. If the cell is totally clear, cell is totally clear, we also can divide like this. Okay, agranulocytes. Agranulocytes do not have specific granules. Okay, do not have specific granules. According to this classification, all the lymphocytes do not have granules. Monocytes, this cell belongs to myeloid line but not having any granules. Okay, lymphocytes and monocytes. So these two cells totally clear. These two cells, one have much bigger nucleus in center, another have C-shaped nucleus like this. Okay, this is a monocyte. So monocyte and lymphocyte, they not have any granules. Understand? They not have any granules. What are the granulocytes? The granulocytes, they contain specific granules. If they having granules, basophilic granules, the some having eosinophilic granules, the some have neutral granules, but they have granules. Basophil cell, basophilic granule, eosinophil cell, eosinophilic granule, neutrophil cell, they having neutrals. Okay, and another important thing,